Today we will again start off with quantum sensing, but an entirely different topic than yesterday. Yesterday we discussed atomic gradiometers to measure gravity, together with Nicole Metche. Today we will discuss an entirely different type of quantum sensor, namely a quantum magnetometer, used to measure magnetic fields extremely sensitively, so sensitively that it can even be used for brain imaging. We have the best speaker for that purpose with us here, Svenja Knappe. She has been pioneering this topic for 16, maybe even more years, first at NIST, the National Institute of Standards and Technology. She is a research professor at the University of Colorado Boulder, and founded her own company, FieldLine, where she is the Chief Technology Officer. FieldLine is commercializing a functional brain imaging system based on quantum magnetometers. And today, Svenja will introduce us into this type of quantum sensor and how it can be used. Svenja, please take it away. And everyone, please ask your questions on Discord, and we will ask them to Svenja after the talk. Thank you very much.
All right. Well, thank you for the kind introduction and also for the invitation. And it's a real pleasure to be here and see how many people have participated over the past days and are participating now. And so I want to talk about translating some of these quantum magnetometers or atomic magnetometers into industry and into real world applications, specifically in an application of brain imaging or functional brain imaging. Here on the right, you can see one of those systems we've been building, and it has about 130 of these magnetometers on a helmet to image brainwaves of a person, in this case, this child. But let's step back and see why we might want to do this. So my research has really focused on the question of how can we take these lab systems that we build out with optics on optical tables, lasers, detectors, gas cells, and can we translate that into devices that people can use in everyday applications. And so we need to develop setups that can be miniaturized and translated, but we also need to develop enabling technologies for manufacturability to produce large volumes of these sensors, usually at fairly low cost, at least for an atomic physics experiment. And then also, how do we package these sensors and make them low size, weight, and power? And this was really pioneered by DARPA in the chip-scale atomic clock program in 2001, where it was really the first time where microfabrication technologies were used to take a very simple atomic vapor cell spectroscopy setup, so an atomic clock, and translate it into a package and then into a commercial product. And here on the right, this was the product that was commercialized by Symmetricom, and then they subsequently got bought by Microsemi and Microchip. And so the question is, how do we do that? And leveraging microfabrication enables us to do some of this, of producing these sensors at large volumes. Why do I want to use atomic sensors? And so there are lots of reasons. So this was an example of an atomic clock, but it could be a magnetometer, a gyroscope, accelerometer, Rydberg sensors. So there are lots of quantum sensors that are used in applications and can push applications forward. And so for some sensors, the reason is really precision. I can get very high signal to noise with these quantum sensors as compared to standard sensors. And often they're really only rivaled by superconducting sensors to get the same performance. But sometimes it's also other reasons. For example, long-term stability. I don't have to recalibrate these sensors, so I can get high accuracy. And so we really have to look at the specific application to see, do we have an advantage of using quantum sensors in these? Why would we want to do that? So here I specifically want to talk about brain imaging. And to set the stage for that and understand why would I want to do that? Is there a market for this? Is something like this needed? I want to give you a very high level idea of brain imaging technologies currently. And so, but first the question is, well, why do we want to image the brain? We have lots of brain imaging modalities. Don't we know a lot about our brain? And we do. But there's also a lot of things we don't know. And that's, I think, really fascinating. At least for me, this types of brain science is very fascinating. And so the brain is a very complicated system. And in a very complicated system, it's very hard to understand, and a lot of things can go wrong. And so if we think about the brain, there's actually a lot of things we don't know. And so some of those questions that I rather find fascinating is very basic questions about the brain. How is information encoded in the brain? We know that in response to a stimulus, like a flash of light, we see neurons fire. But how are more complicated processes encoded in the brain? And there's a lot of open questions still, how that is specifically done. How is memory stored and retrieved in the brain? How can I access memory fast? We know it has to do with synapses, but how are really those mechanisms of finding the information I'm looking for? Some basic questions like, how do we sleep? In the recent years, we have made a lot of progress of understanding why sleep is so necessary, why if you deprive animals or humans of sleep for only 2 weeks, they die. And so it's a lot of the cleaning functionality, but we really don't know the details of it yet. But it's another basic question of understanding what's going on and what do we know about the brain. Questions like, why do we laugh? Why do we dream? What's going on when we dream? How is time represented in the brain? And then this question of how much of our personality is determined by our brain. I think usually we think the brain is kind of the central organ that defines us as humans, and what defines our personality, and also this question of, is there a free will? I think most of us think yes, we certainly hope there is, but then where is it? Like, why haven't we found what it is, this thing we call the free will? And then also, what is consciousness? This is really how we define ourselves as humans, right? Consciousness. And the question is, what is it? And how is that representation in the brain? And so there's a lot of things we really, really want to understand about ourselves. And so there's this natural curiosity of understanding how do we work? What defines us as humans that we really don't know? And so the brain's a complicated structure. That's why we haven't really understood those functional interactions. But then in a very complicated system, a lot of things can also go wrong. And I keep showing this picture because it was kind of one of those moments in my life where I looked at this sign, which I found in Australia, in the bathroom in the airport. And it says 1 in 5 Australians are likely to be working with a mental health condition. And I thought, oh my God, that is huge. 1 in 5. Count how many people are on this call. Well, 1 in 5, right? That's big. And I thought, oh, okay, so Australians are definitely working with a big problem here. But then if we look at the statistics, this is for the US now, it's exactly the same number. It really doesn't matter if we look at Australia, if we look at the US, if we look at Europe. 1 in 5 people suffer a diagnosable mental disorder. And if you want to look at the definition of diagnosable, you can look at the reference I put in here. So this is a big number. And so this is a big problem for society, not just for people that are affected, but also the people themselves, but also their loved ones. And so apart from this natural curiosity of understanding who we are and how our brain works, there is really something we need to address and we want to address. And then, of course, we can turn this into an economic burden. That's kind of the commercial side to things. Beyond the scientific side, the cost of health disorders are about $1 trillion for the US, and it's this pretty much identical number for Europe, and $16 trillion globally. So that's an enormous number. And if you look at the main disorders here on the right, you read through them, a lot of these are diseases that don't really kill you, but that make your life miserable for a really, really long time. And that's why this number is so high. And so there's also an economic reason why we might want to address better treatments and diagnostics, differential diagnostics of brain disorders. And so in a lot of these disorders, we really can only treat the symptoms right now. We can't really treat the underlying disease because we don't know enough about the disease yet. And so there's a big push for, or a big need for improving our non-invasive imaging technologies of the brain. And so if we look at brain imaging modalities, so mostly I focused it on functional brain imaging modalities, except for MRI here. And this is temporal resolution on the horizontal axis and spatial resolution on the vertical axis. I think everybody has heard of MRI. It's really the workhorse of brain imaging, but it's not really a functional method. It images water distribution, or proton distribution. So it's really water distribution in the brain, and it gives a really high contrast for morphology. Where are things? And sometimes we can see things like tumors really well because the morphology changed. But in a lot of these functional methods, the morphology hasn't changed. Something else changed in the connectivity or in how these waveforms, how things function in the brain, but really you can't see it from this water distribution. There's a method called functional MRI, and it really measures oxygen distribution. It distinguishes between oxygenated and deoxygenated hemoglobin. That gives the contrast in fMRI. And the spatial resolution isn't quite as high as for MRI, but it measures functionality, because usually for function, you need oxygen. And so it's an indirect method, but it can tell us something about function. Then there are the radiotracer methods, such as positron emission tomography or SPECT. So there you inject a radiotracer that's bound to glucose. And so you measure glucose distribution, so sugar, because again, that tells you something about activity, but it's also an indirect method, but yet another method that can give us information about this whole picture. If I'm interested really in neurophysiology, what's really going on with the neuron directly, I can use electroencephalography, so I can attach electrodes to the head and I can get those signals. But you can see that while the temporal resolution is really high, the spatial resolution isn't, because those signals have to travel through the skull, and so conductivity matters and distorts the signals. If instead I can use magnetic fields, the skull is transparent to magnetic fields, and so with an array of magnetometers, I can pick up the magnetic fields outside the brain. I get slightly higher spatial resolution, and again, axial temporal resolution. But if I want really, really good temporal and spatial resolution, and when I say really, really good, I'm talking about millimeter spatial resolution and millisecond temporal resolution. The only method right now is really to drill a hole into the scalp and stick electrodes into the brain. And that's exactly what's done if I am trying, for example, to localize an epileptic seizure or something like that. I want to know where's the focus of that seizure, but I do need to insert electrons into the head. And that's, of course, neither pleasant nor really good in terms of morbidity. So hurting the patient in the process. And so one of the goals is really, well, what if we could use this non-invasive method of MEG and move it to higher spatial resolutions and temporal resolution? And this is one of the quests of why we might want to use these quantum magnetometers. This is how an existing magnetoencephalography system looks like. They exist in hospitals. And so here, this is essentially a ginormous liquid helium dewar, because they're low temperature superconducting magnetometers embedded in a helmet that sits on the head of this patient sitting inside, and you stick your head inside, and then your brain fields, as they come out naturally out of your head, are recorded with this large array of magnetometers that are embedded in this dewar, and then you can reconstruct where the sources came from. And so you get decent spatial and good temporal resolution, but it's very large infrastructure. It needs constant upkeep of liquid helium, which is expensive and harder to get. And it's very fixed and it's very expensive, which is really also limiting the distribution of these systems and how many systems there can be in the world. And so one of the questions is, well, if I can make small quantum sensors and I can move them directly on the scalp of the patient, what could I do with that? Is there really a market? Is there a need for this? Can I improve brain imaging? And so one thing you can already see, for example, you have a little child here, and you can see this really big gap between the head and that stationary helmet. So brain fields fall off in the far field as R square. So they're already really small when you're right at the scalp, but they get even smaller as you go further away. And so these big gaps cause an issue. The fields are pretty much too small to pick up, but also this child is definitely not still. It's not going to be a nice and still while you image it if they're going to bobble around in this helmet. And so you have to deal with lots of motion artifacts. So the question is, well, can we do something for brain imaging if we can move these sensors closer to the skull? And so this is a simulation study by a group in Nottingham, and they have calculated what is the signal to noise ratio if I move those sensors closer to the scalp and use quantum sensors as compared to existing MEG. And so the noise floor of our sensors isn't quite as high as it is for the squid magnetometers right now, at least the commercial version. But if you look at kind of signal to noise on the left there, everything is yellow or red, and then the deeper structures are blue. So you get an improvement of signal to noise of the quantum technology as compared to the current technology. And so signal to noise goes somewhere between 2 and 10, should increase. And then if you look how does that translate into reconstruction accuracy of those brain sources, on the right, again, is the conformal MEG. So those are the quantum sensors. And in the middle is the existing technology. And you can see on the right, everything is pretty much red. So I can get higher reconstruction accuracies with the use of this quantum technology. And so this is really promising. And there have been 5 other studies by now that calculated similar things, and they predict similar, they all agree in that you get better reconstruction accuracy, or you can get higher spatial resolution, depending how you define it, and also higher information content if you manage to put these sensors on the head. But all of these so far have been simulation studies, but it's very promising for technology to build. And so experimentally, we haven't solved this quite yet, or nobody has really solved it quite yet, but we and others are making big progress in that direction. And so what is really the advantage of using these quantum sensors? We call them optically pumped magnetometers, OPMs, for this brain imaging method. And so we just said, yes, higher spatial resolution, higher information content, but then also it really a better fit for people with non-standard heads. This one size fits all helmet only fits very, very few people well. most people could fit inside, but there's gaps somewhere. And so imaging brain sources independent of head size and shape, because most of us don't have that standard head. Studies in children, we've already mentioned. Now the sensor moves with the person. And as you image the brain, you can start going to much more naturalistic paradigms. You can start measuring while people interact rather than being stuck in a big machine as you try to understand more about the brain. Maybe at some point we can do it through telemetry. We're not quite there yet. We still need a shielded room, just like MEG does. But maybe at some point we can move there. And then the hope is for less expensive systems, especially also less expensive upkeep, to really democratize this field, that is available for a much larger fraction of our population. And then you can start doing new studies, more interesting sleep studies, studies maybe of several people interacting, understanding what's going on in the brain there. And so I think it's a really exciting technology that could really enable a lot of new studies in the field of brain science. So let's talk a little bit about the technology before we run out of time here. The first MEG signals were measured already in 2006 by microanalysis group by using a gigantic glass cell and holding it next to the head, and you can see the first brain signals here. And there was lots of questions of where this really brain signals, and what can you do if you can really, instead of one big glass cell, put a conformal system that you can put all around the head. And so this was how the magnetometer signal looked like. You had a vapor cell here, and you shown a circularly polarized pump laser in one direction. And then you had a probe laser, and you looked at the polarization and rotation of the probe laser and detected if the balanced polarimeter. But if we want to build 150 of these and put them on somebody's head, we want to make them as simple as possible to really make them uniform and at low cost. And so the first thing is, well, let's shine pump and probe laser from the same direction. Let's simplify the system as much as we can. And then, well, let's rather use one laser. I don't want to do separate pump and probe, too many resources. And then the question is, well, instead of a polarimeter, can I just work with a simple detector? And this now looks like a really, really simple system. And we managed to retain a lot of the sensitivity and performance, despite the fact that we made it really, really small. And so what I'm looking at here is really a zero field resonance as I shine a circularly polarized laser through a vapor of atoms. And at zero, I can see this line shape. That's not a very good line shape to work with. So I need to put coils and then use lock-in detection to get higher signal to noise and also get a nice zero crossing. And then I can lock my, my coils onto the zero crossing. And so now I have a magnetometer that actually works in this spin exchange suppressed regime. I haven't really talked about it, but gets very high performance because I'm working in a regime where I have lots and lots of atoms. And it's a direct field magnetometer. I'm sensitive to the direction of this applied magnetic field modulation. But it only works at very low magnetic fields. That's really the caveat of that. So for this type of magnetometer, I have to keep working in a shielded environment. If I really want to fabricate this, I need to develop a lot of enabling technology, miniaturize. So I need small lasers, I need small vapor cells, I need small packages, and put together a physics package that's very small and integrated into it. And so the first of these sensors were already built in 2009. We built two, and we were lucky that, so at that time I was still at NIST when we developed, and we were lucky to work with the PTB in Berlin, who know how to do brain imaging and who were very patient with us and really followed us through moving this technology and also validating it against existing brain imaging systems and telling us what we need to develop. So those were really our customer feedback. So we developed towards something that's helpful in this field. And so over the years, we have developed many of these different systems, and we improved the performance, and then we actually moved it into the commercial range in 2017, because there was really a customer pull. People were asking us, can you build this for us? And we said in research, no, we cannot. We can't build you 200 sensors. But in industry we can. But this is really a process where you develop technology, but you're also educating the people in the brain imaging field and validating it against current technology. And I think this is a really important cycle that we need as we translate quantum technology into applications. We need to make sure we translate them towards something that's needed and have this intimate cycle of users and producers and developers, and then also educate that field, because it's good that we had PTB, but it's only useful if the whole field of MEG is educated too and understands what this new technology can bring them. And so this was a really important cycle to go through. And so these are the recent results. Now, if I take this commercial system, 128 channels, and I measure the response to a beep in the ear, you should see a response in the auditory cortex. And so on the left, you can see it with the quantum sensors, and on the right, you see it in the same scale with the existing technology. And you can immediately see that the amplitudes are a factor of 5 at least higher. And so the signal to noise is also higher. This is exactly what those theoretical studies predicted. And then you can measure in all over the head, and you get a heat map that's kind of on the bottom left. You see a dipolar field in the auditory cortex, and then you see a response in the other auditory cortex. And so the signals look like a dipole, and then you can reconstruct where the activity comes from, and that's kind of the yellow activity. It overlapped with an MRI of the person, and you can do that in a time-resolved manner. And so we need to develop technologies for that, and really a whole medical product around it that makes this useful in addition to just the sensors. So then you could start working with people on these medical applications. And so let me finish there. Of course, this is a whole team of people that have been working on this, and I'm just going to leave this up in terms of names because I think I'm out of time.
Wonderful. Thank you very much, Svenja. This is really inspiring topic with so many different fields also involved. Physics, psychology, neuroscience. You need electrical engineers and also mechanical engineers to build everything well. So this is really inspiring. We have many questions. I will start asking them. First question is about another type of quantum sensors for magnetic fields, namely NV centers. The question is, could NV center ensembles give a better spatial or vector resolution than OPMs, the optically pumped magnetometers that we discussed today?
Yeah, thank you. So this is definitely my personal opinion that NV centers, they do get higher spatial resolution as a sensor, and they have some advantages in terms of being vector sensors. For brain imaging, I don't see it. I don't see that the spatial resolution of the sensor will give better brain images because I'm working at a certain distance. So it's really noise floor that's determinate for this type of brain imaging. For imaging other things, absolutely, but I don't see it for this type of brain imaging.
Then, is there a role for quantum computing here? And could there even be an advantage of embedding computational qubits in the quantum sensor?
So maybe. So I could see the quantum computing right now more on the analysis side. Like this, again, complicated system. And if we find the right question, that's where quantum computers could shine. In terms of embedding something into the sensor, I think I try to relay this idea of we need to make it as simple as possible because we need to make so many of it. And a lot of we can improve the sensor in lots of different ways, but usually at the cost of making it more complicated or more expensive for a small gain. And so I think as we translate these technologies, we need to be conscious about how much are we going to gain by adding things. I mean, people have shown improvement of spin squeezing and things like that, but the complexity that's added isn't outweighing the gain right now. And then maybe my sensor gets bigger and then I can't put as many on there. So again, we're trying to weigh, and at least I can't see it immediately how that would help on the sensor side right now.
Shika asks from the US, what problems can theorists, especially physics theorists, contribute to bring these quantum sensors closer to even better practical use?
So right now I can see a lot of need on the analysis side. Like so you're picking up brain fields in lots of locations and you're trying to understand something about a complicated system. So that's definitely one side where I think theory is needed and really helpful to understand these underlying brain mechanisms. I also can think that this is a very simple sensor right now from a physics perspective, that we can make better sensors, absolutely. And so understanding how, without making it way too complicated, I think is an area where there's a lot of things we still don't really understand, even though the sensor is really simple. How can we understand these systems better, and how can we improve systems on the physics side of things?
Some people are already enthusiastic to help working on it. Juliana from Italy asks, do you have an open data set with quantum sensor acquisitions in order to develop machine learning processing or imaging procedures for this type of quantum sensors?
We definitely have data sets, and we're happy to share data sets with people. Send me an email, and I can tell you specifically what type of signals we have. And if you have something specific in mind, we also have systems that are open in the sense that we can take other data sets if there's something specific. We're very happy to work with a community of really making these systems useful.
Amazing. And to share from the WOMANIUM side, this could definitely also be one of the global quantum projects that we have at the end. You're totally free to choose there what you do if you pick the open project, and this could be part of it. You say it's a simple sensor, but for the non-physicists, it's not so simple. So could we take 5 minutes to go through your explanation again of how it works physically? What is the difference between a pump and a probe laser? What is really the take-home message here for how this atomic vapor cell-based sensor works?
Okay. Sorry for that. I have to go back and find the... Okay, so what's really happening is I have a laser and I have a container of a gas of atoms. And so what we call cell is really that container that houses rubidium atoms in a vapor. As we heat it up, we get more and more atoms in the vapor. And then the laser is on resonance and it's circularly polarized, so it carries angular momentum. And it has a spin that carries angular momentum, and it's transferring that angular momentum to the electron spin And so it's pretty much like little magnets. They're starting to orient themselves inside that vapor cell. And then in a magnetic field, they will start precessing around that magnetic field. And so what happens is that when all my atom spins are pointing in the direction of the laser beam, I can't transfer any more angular momentum. It's all transferred. And so the light will just pass through the cell. But if there's a magnetic field, those spins start orienting themselves slightly differently. And so now I can transfer angular momentum again. Now the light can be absorbed again, so more angular momentum can be transferred. And so if I look on this magnetic field versus transmission signal, this is really the light that comes out when I shine my laser through. If I'm at zero magnetic field, very little light gets absorbed. So most of the light passes through, gets on my photodiode. But in the presence of a magnetic field, those spins are slightly differently oriented and they will absorb some of that light. And so that's where I get this resonance. So it's really a magnetic field to optical transducer. That's what this is. So the amount of light at the end, at least when I use my magic with my coils, is proportional to the magnetic field. I hope that helps in a very hand-wavy explanation of how these work.
Definitely, that is great. And Svenja asks, why rubidium atoms? Could other atoms be used too?
You can absolutely use other atoms. Pretty much any alkali atom can be a good choice. These magnetometers have been built with cesium. They've been built with potassium. They have been built with helium. All of those have slight advantages and disadvantages. And so now we're getting into the kind of details of why would you pick one versus the other. And that really has to do with noise floor and also laser availability. Like where can I find good lasers that are inexpensive enough and high enough reliability, but other atoms are definitely, I'm sure there's even more atoms, but finding an atom that again works at decent temperatures with the right laser sources and gets good performance is really what you're trading off.
Prashant asks, how does the performance compare with the performance of a squid, especially the sensitivity?
Yeah, so the commercial squids are around 3 femtotesla per hertz noise floor. The smaller ones we're building, they're usually under 15, maybe as good as 7. So somewhere there with the current, like, so the size is 3 millimeters by 1.5 by 1.5. So about a factor, I would say, of 3, or maybe 2 to 5 worse currently than the low temperature squids that are in the MEG systems, and their pickup coil is, let's say, about 1 centimeter to compare to that 3 millimeter size. So, yeah.
Yeah, she's concerned if the sensors harm or disturb the brain in any way with all the lasers. I think this is all contained in the cell. Is that right?
Yeah, so this is really like this sugar cube package. And so first, these are VCSEL lasers. So the power is very, very small, like 200 microwatt. So less than probably your mouse, like the red laser mice we have. And second, they are completely contained. And you're right. If you do build a medical device, this is a question, and you have to make sure that these are safe and nothing can get out, and we have to heat the vapor cell and the person can't get harmed. This is a technology, though, that does not introduce energy into the brain. Like, so it does not, like MRI, that where the question is, do I heat the brain? Do I actually cook the brain? So this is a very passive method, but yes, absolutely, in a medical device, you need to ask those questions. And we need to make sure that as we build them, we're not harming people.
I will ask two last questions. I will ask them both at the same time. They are both about applications. Dawal from India asks, can we do any analysis with this instrument for an active person? For example, playing a sport, playing video games? And then Artem from Mexico asks, can the data be used for simulation of processes in the brain, like simulating the neural activity of a person in the state of sleep?
So we can definitely use them for active people. One thing that I think interesting is brain-computer interfaces. So kind of a feedback loop of using brain waves to steer things, or to play games, but for example, also to for amputees, right, to steer prostheses or things like that, or also learn more about the brain, again, to maybe steer brain stimulators, and that way close a loop and help people, or for stroke rehabilitation or things like that. But also, I think, one area that's very close to sport is traumatic brain injuries, like that happen often in sports. But I'm sure there are people that can spin up a lot of applications in those related fields. So this is a really big question, so I'm not sure I'm answering it in a useful way. And then learning something about brain processes. So right now, you cannot image single neurons. You image clusters of neurons. This technology, though, can be scaled to different sizes too. So we, for example, image the brain of rats, so small rodents, or you can get even closer to brain slices where you can start thinking about imaging very small structures, and using these at all these different size levels will probably help understanding how neurons interact, or can interact, and then how that translates into brain regions interacting. But we cannot image single neurons with this technology. I at least want to make that really clear. We're not trying to, at least not inside a human right now.
There are lots more interesting questions, Svenja, but we need to wrap up now. Thanks to all participants for all the great questions, and special thanks to you, Svenja, for this great and inspiring presentation. I encourage everyone to keep thinking from your perspective and your field. What can we do with these sensors? What can you do yourself to improve the data processing, or come up with even better fabrication methods? Svenja, thank you very much. And we would love to host another session with you to go even more in depth and answer everyone's question. Thank you.
Thank you. It was a pleasure.